I am so thrilled to introduce our next speaker, Chris Mason. So Chris, I've known Chris for a couple of years now in a variety of different settings. And as you will all see, the density of information that Chris will share for you in this period of time that we have, it may be the highest density of information share of like any person that I know. So just open up your brains a little bit because there's about to be a landslide of just amazing work and projects. And I know, Chris, your lab has been doing some really sensational work on diagnostics and other work that also very much involves community and grassroots engagement. So we're so thrilled to have you. I know this is your first time speaking at BioSummit and being a part of this community. So really just an honor to have you. And everybody, can you please join me in giving some love and some snaps and some emoji love for Chris Mason. So Chris, thanks so much for being here.
Great. Thanks so much for having me. Can you hear me okay? Everything coming through all right?
Yes, we got you.
Great. Wonderful. And it's an honor, a pleasure to be here. Always great to see you, David, and great work on really leading the effort to get this together. And we've done a little bit of work here and there to build communities, and it's always a lot of effort, but also a lot of great reward when you get such a global group like you've got here. So it's my first BioSummit, so I'm excited to be here. But I've heard about it, seen a lot of the work online over the years. And let me share my screen, and I'll describe some of the work we've been doing in lab. I also just tweeted out my slides. So as David alluded to, I usually have a lot of information, but we're a little bit behind time. So I'm going to try and go even extra fast and see if we can get back to try and finish at 10:15. You think you want to, or 10:10? What was it?
10:15 is perfect. Yeah, 10:15.
So I'll leave it for Q&A as well. So I'll walk everyone through a variety of slides and data, but again, just tweeted my slides out. So don't feel like you have to take notes. I'd like to make as much of my science open as possible, unless it's under a strange, mysterious company in the Cayman Islands. I don't really have any Cayman Island company, but if I did, that's where I think secrets would lie. But in any case, I'm going to just go through a range of high throughput tools for responding to COVID-19. I think all of us know, the breadth and sort of damage of this crisis really globally. So I won't belabor that point. I'll focus just in particular about when we got hit really early, just in New York City, where I'm from and where I am now, and originally from Wisconsin, but now I live in Brooklyn. And we really wanted to start to better understand the virus and think about why is it hit so hard here so early? Why is it still ravaging? Is it changing? Is it evolving at all? We just got some data from the hospital on Friday that indicates new mutations are emerging. So it's a constant battle and constant challenge. And when it first kind of broke, I realized there are a lot of ways you could look at the virus. And so I wanted to put this slide up first. And what I'm going to do today is walk you through, what are the ways you can study both the virus as well as the host response and better understand even what happens when the virus rages through the body and what does that do to the body. So, we'll focus first on some of the RNA sequencing methods that we've developed over the years with the FDA and with Illumina and other groups, and how it's expanded into other directions soon, because this gives you both the host and the viral RNA. So in particular, paper that we have that's in review, it's also up online as a preprint, is an examination of the first 735 patients that came through, and we did some of these samples in duplicate and sequenced basically out these samples with total RNA-seq, meaning all of the RNA that's there, we capture it and sequence it after we've done a ribosomal depletion. So it means anything that's not ribosomal RNA, we take everything left over and sequence it, compare this to RT-PCR, and then compare this to LAMP, which I'll tell you about at the end. And so I did this for all the patients who walked through the door, and of those that were actually positive by RT-PCR, is about 216 patients, and some were negative, but we learned a lot from them as well. And then we also started looking in the subway, which I'll show you some of that data. I have a real penchant for swabbing environmental areas, and I'll come back to that also in the talk today. So one of the things that's really interesting about it, if you just take all the RNA from a nasopharyngeal swab, you grab it, you sequence it, you can take all the reads and map them to their provenance, where they came from. And you can see your human RNA is in blue, bacterial RNA here is in green. And you can also see in some cases, we'd see spikes of this viral RNA, SARS-CoV-2 here in orange, sort of high, medium, or low. In some cases, there were false negatives from RT-PCR, but we could still pick them up with sequencing, and some patients had really high viral titers. And so what's exciting about this is you can take the fragments of RNA from the virus, map them back to the Wuhan strain, and then start to call variants. And then you can trace the evolutionary history of the virus. You can see here, any dot is a difference, a mutation relative to the Wuhan strain. So we could see all these different mutations and put them into clades and start to build out sort of a phylogeny of where we think the viruses came from and how they're related to each other. And so in particular, we could pick up things like a deletion here. This is compared SARS-CoV-2 versus the SARS-CoV from 2003. You can see the two coronaviruses are highly conserved, but you can pick up deletions here. And what's really exciting about this is not just trying to think about, okay, what does this deletion mean? But because everyone has been sharing so much data so fast with all their viral sequences, we were quickly able to see that we weren't the only ones that could pick up this deletion. You can see here's our deletion on top. And here's Reykjavik, here's Australia, here's England, Ohio, Washington, all the different places that carry this mutation. We could quickly pick it up and validate this in other cohorts, which is very beneficial. So we could bring the data back in and then compare again, okay, do we have our data from Cornell and New York Presbyterian versus other New York City? We could see this enriched clade of these mutations consistently showing up in the early part of the outbreak in New York City. And then we can compare this to what do we see elsewhere in the world. So if you look out, this is the 20C clade, you can see New York, it was the predominant strain in New York City, much more so than these 4 other clades. And if you go down this way, you can see the East Coast, Midwest, West Coast of the United States, and then Asia, Europe, Oceania, Africa. We can see this really was kind of a unique feature, especially in New York City, of having so much of this subclade. Now, it's enriched for the D614G mutation as well, so it may have some clinical relevance, but so far, it's not been borne out by the clinical data. But it's interesting to note that we see these different clades so far being differentially present around the world. The other thing we looked at is, okay, well, that's going to from the patients. What if we look in the room in which the patients are sitting? So we actually took areas where the patients were sitting, hot zone, patients near the areas near them, and a place where in the hospital where there weren't any COVID patients. So we could see difference of the viral density in these different areas of the hospital. So if you look at this visually, we could see the floor of the patient rooms in the hot zone where patients were sitting, the sort of equipment, the railings, even the curtains, even the bathroom. You can see here the toilet handle and even the toilet ceiling had small detectable amounts of the virus. We did culture this. We don't think it's infectious virus based on infectivity studies. So we think it's just the remnants of the virus, but it's floating all over the room, not as much present in cold zones. But here too, when we sequence the data, we can actually reconstruct the whole sequence of the virus from the walls of the hospital, and then also infer the likely origin and phylogenetic relationship of each one of those strains. So this work is also in review now that lets us better understand, what's in the world around us. And so then, of course, this leads to the point I brought up earlier as well. What about in the world broadly? We have a large consortium called Metasub, which is a metagenomics of subways and urban biomes, where every year we normally take samples as more of an exploration effort. But in March of this year, we began to sample for COVID. So we sampled in public environments as well as in hospitals to see what we could pick up for presence of any RNA viruses. So in particular, we did pick up some, human RNA and a lot of unknown RNA, which is, I think, kind of exciting, actually. I'll come back to that. But also influenza, we could pick up other even plant viruses. But in our first test in New York City, we didn't see any coronavirus. But since then, we've now gotten enough data back in the past few months where we can see different densities in the different cities we've sequenced so far. And we're in the process of finalizing and validating a lot of these with RT-PCR. But we do see certain sites in the cities that when they were having their outbreak, we can pick up some of the virus. Depending on where you were at which time. And so this is interesting to see that, this gives you an environmental way to actually characterize the world's virus very quickly with sequencing data. And it's not just on its surfaces. I don't know if any of you remember, but there was a headline that the Bronx Zoo tigers had coronavirus, which I think when this happened, everyone probably looked at their cat in their house and said, you looked at them maybe a little bit funny. So we've started a project to figure out how common cats and their owners carry a virus, and then also what other viruses are present. This is through Metasub, a project called MetaCat. So we actually just got the IRB and iCook approved last week. So if anyone wants to, you can now get a stool, get a free collection kit. We'll send it to you, and you can collect a stool and mouth sample. It's up to you how you convince your cat to give you a mouth sample, but a stool sample is pretty easy to get. So if you'd like to contribute to the study, please do so. Let me know if you have any issues. Okay, so that's the coronavirus, but of course there's other viruses present. Trevor Bedford is one of the, Trevor Noah is great. Trevor Bedford is one of the great Twitter sort of digesters of viral information. He's fantastic, and the Seattle flu study has been tracking other viruses for a long time. We see the same thing in our data from shotgun RNA sequencing. We can pick up here in pink other viruses present in the low, medium, and high patients. These are ones that had confirmed COVID with SARS-CoV-2, but you can see the people who came in, they all thought they were sick with COVID. They met the diagnostic criteria, but these people on the right over here all had these other respiratory viruses. The other thing we could see, that was on the viral side. If you look at the bacterial side, these are normal flora bacteria, normal microbiome that's present. And in the high titer patients, when we go back, high titer patients, we can see there's this depletion of a lot of their normal flora. So there's another interesting benefit of the shotgun method is you see bacterial, viral, human, any sort of RNA that's present, and you can look for disruptions, in this case, also in their microbiome. So to that end, like I said, it's all kinds of RNA, including human. So then we looked at differentially expressed genes, or DEGs. These are the genes sorted by the fold change, the degree of change, and the statistical significance here on the y-axis. So these are genes you'd expect to see that are interferon-stimulated genes or related to viral infection. And if you group these into a pathway and say, what are these types of genes, you can see here they make sense. You can see interferon alpha and gamma response, but down here, really surprising things. We could see olfactory signaling or hemoglobin or iron binding. These are sort of the features that define the cardiac and hematological phenotypes. And here, the fact that people lose sense of smell and taste, we can even see that in the data from the nasopharyngeal swab. So really interesting to see that. And then we made it up so you can make these plots. So if you want to have like a heat map of different cytokine and interferon signaling, you can see this here. And we've also built up a website. So if you want to make your own plots, here, I'll do a quick, I'm sharing my screen. If you go to covidgenes.weill.cornell.edu, we built a nice interface so you can make your own heat maps just like the ones we put into the paper. So if you'd like to pull them up, it's a open forum and portal, so you can basically make violin plots of any gene in the genome and see what's the difference as a function of expression, or you can do multi-gene heat maps and plot them, or you can do pathway enrichment as well. So built this as a public resource to help anyone who's looking at specific human response genes, and that's online as well for everyone to use. And we've used this already to look at complement factor, which we know was a function of clotting issues in some of the patients, and then also just published this a couple months ago. So we know that is a factor for sure in the infection. Okay, so that's the RNA-seq set. I'm gonna switch gears in the next 8 to 9 minutes and talk about spatial transcriptomics, which we've been doing also with the NanoString geomics platform. So lets you look at one slide, basically UV cleave oligos to look at a region of interest, and then we can sequence everything that's found on this slide. So you bind antibodies to it, grab them out, and sequence the tags. And here we can actually see that TMPRSS2 and also ACE2 was expressed pretty high in the alveoli. You can see here, these are the tags. Blue is for DNA staining, dapping. You can see the tag for the virus and also the ACE2 and TMPRSS receptors. And we can see this is actually, these are autopsy patients. This is after the patients have died, unfortunately, but the virus is mostly gone, but the damage is still there. So actually the virus cleared in some of the patients we observed, but it was still too much for the body to recover from. So in particular, some of these alveoli were completely crumpled and collapsed. You can see it in the lung tissue. And the other thing that we observed is you look at the types of cells that were present. This is basically COVID flu versus normal lungs in the alveoli, and then acute respiratory distress syndrome. You can see your differences. But look here, this orange is a huge infusion of macrophages that came in, didn't leave the lungs and ended up leading, probably contributing to this patient's death. In other cases, we could see a large number of neutrophils also invading that are, unexpected, too high, and too much for the body to deal with in terms of that sort of immune infiltration. And this other large airway vascular, other control samples over here. So that's, other ways we're looking at the patient samples, and there's an autopsy project we're in the middle of finishing up, but we also want to get faster ways to do sequencing. And so this, I think, is very relevant for a lot of what the bio community does. And also Jogl has been a really great collaborative effort with all the people in Jogl and people sharing methods and protocols and techniques. And so in February, we started working a lot with LAMP and saying, okay, well, could we use LAMP? And this is a loop-mediated isothermal amplification method. It's kind of a cousin of PCR where you have 6 primers instead of 2, target one piece of DNA or RNA, and you convert RNA into cDNA and then make that into double-stranded DNA with a strand displacing polymerase. Worked a lot with New England Biolabs on this very early on, and I started to work on the protocol. They shared their primers and said, okay, this is great. Let's give this a shot with samples in the lab. We brought it in, started to look at controls, and the reaction starts all pink, you can see, but as soon as you have different copies of RNA, it starts to turn yellow, being so colorimetric shift, and by 30 minutes, you can often get between 10 or 100 copies picked up, and it's that easy to see, is the virus present or not? You can just see it by eye. And what's really exciting is we then tried clinical samples at the end of February and early March and realized this was working. It's like, holy crap, this looks like it's that easy. You can tell just by eye. We ran a large set of clinical samples compared to RT-PCR and RNA sequencing. And you can see the third dimension here is the LAMP, the intensity of the LAMP reaction, which you can see was pretty well correlated. And then we started doing about over 200 clinical samples. We could see here we had pretty good specificity and decent sensitivity. And then on the very high CT values where it started to lose a little bit of it, but overall across all the patients, we did look very good. We then, instead of putting one primer target in there, we did 2 sets of primers, including the E and N genes. And there we can get reliability down to 25 copies of the gene and sometimes 5 or 10 and a lower end of sensitivity. So we're really excited about this. Open up the protocol to anyone we possibly could. I was kind of screaming it from the rooftops. Worked also with color genomics in the early days. That led to the first FDA EUA authorization for this test, which is now being used to screen about 10,000 patients per day in San Francisco. This also New England Biolabs, and we worked with them very closely with Jogl as well as to help test and benchmark a colorimetric kit. So now you can just buy this as a kit if you want with all the controls. That's fully available. And also we made a global group. There's a global LAMP consortium, or GLAMP, which is kind of a joke about glamping. But this is, again, an open group, open consortium of everyone working on automation, testing, variations of LAMP. It's a pre-competitive space. We want people to share ideas here. Eventually, maybe they'll get commercialized, but we really want it to be open and shareable with everyone. So it's now we have calls every Thursday at 12 o'clock Eastern to share methods and have talks and discuss this, also with a lot of Joggle members as well. So we took this and said, okay, this seems like it's working. What if we could actually do a pop-up lab, right? This is something we've done before at conferences. We'll go to a conference and swab everyone's phone and sequence it just for fun. And we've done this here. So we thought, well, we should be able to do this, like, for example, in Racine, my hometown, Racine, Wisconsin. Went back there with my brother, who was the mayor. We set up testing kits, got the firefighters trained, and got everyone ready to do their own testing. And this was covered earlier this summer, is that basically, you know, we trained the firefighters to run this test. It's that simple. So now we're expanding this out to other cities across Wisconsin. I just got back from there yesterday, working with also members in Madison, Wisconsin, Milwaukee, members from the governor's office to really ramp this up. And so that's doing it by hand with pipettes, which people can do, of course, but we also wanted to make it easier. So we started to work very closely with David Erickson, Ethel Caesarman at Cornell, also at Cornell. They made a little tiny box. They built this for doing remote testing, mostly in Uganda. They were doing this basically can you just use solar power or butane to run this reaction? And here they also run LAMP, like in this little machine. You can make these basically PCR tubes, a photodiode, and you ramp this up, and it's a small little box that you can basically run the reaction and visualize the results. And so we just have been partnering with Foxconn to manufacture this at larger scale, which is ongoing. But basically, we now have this working in the emergency room. So as patients walk in, we have the tiny box there. We can quickly do a test of patients right when they walk through the door. So it's very great point of care testing, and we're optimizing this. And again, the protocols are deployed online. We want to share this with anyone and everyone we can to make it as easy as possible. Okay, so that's the three methods. I'll close with a little bit of discussion of direct RNA sequencing. That's something we've done a lot on the MinION and also now on the Prometheon, different Oxford Nanopore technology sequencers. And so if you're not familiar with Nanopore sequencing, it's basically a way where you have DNA or RNA here as it moves through the pore, which used to be alpha hemolysin, or it could be CSGG. These are different pores that you can change them out. Basically, as the DNA or RNA moves through the pore, it changes the current, the conductivity of the membrane, and that is commensurate with the base that's going through, as well as the modified base going through. So this has been used to look at SARS-CoV-2 transcriptome. There was a beautiful paper from Cell earlier this year that looked at some of the first data. But I think what's kind of exciting about this, to close on looking forward, is that, you know, we've begun to do lots of deep sequencing of direct RNA sequencing and looking at other sort of variations in the transcriptome, but also taking this to new places. So for example, about 5 years ago, we thought, well, could we just sequence in space, for example, and bring up a nanopore sequencer up there? And so we worked with Aaron Burton and colleagues at NASA and to get up there. That's okay, well, you're gonna need a patch. And said, okay, great, we got a patch, got that set up. Made our way up there, did sequencing in space, got the first genome sequenced and assembled that was not from this planet. We also had the first space epigenome. We published that data last year. And Kate Rubins, you know, she really was an extraordinary leader in this as a trained virologist. She was the first one to do sequencing in space. And I'm really excited. I was just emailing her yesterday. She's on her way back up to space to continue more experiments of direct RNA sequencing and general sequencing methods up there in the space station. So she's a phenomenal collaborator, also super fun and really enthusiast for space and science all at the same time. And I'll close with one last thing, is we do have new methods that we're trying to get out from anyone and everywhere, and that includes the XPRIZE we just announced for COVID-19. Details are here. The prize is already closed for who can enter new entrants or enter new ideas, but it is ongoing. You can see here, we did an analysis of all the different tests on the market and what's their limit of detection over here on the left for this paper, and announced and said, you know, if you're going to start doing pooling or testing, we need methods that are probably more sensitive, but also are cheaper. And so this is the XPRIZE to push on both those directions. And this is now an effort I'm helping to lead as much as we can through the end of the year to make it so there's more testing methods that can be deployed as fast and cheaply as possible. And with it, I want to thank everyone in lab who makes all this work possible and is endlessly inspiring. Thank everyone here who came to listen. Thank, of course, funding. Those who supported directly by World Quant and also Citadel for philanthropic support, also from Bill Ackman, the Pershing Foundation, Gates Foundation, NIH, NASA, and also CTSC. And thank everyone, our collaborators, friends, colleagues, and happy to take any questions.
So has anybody had a chance to breathe yet? Did you take any breaths during that?
Only one breath.
You took a breath at the beginning and then you just kind of went, right? I think that's pretty much what happened. So I tried to warn everybody to open up your brains and make sure that your brains could be filled with the information that was going to be forthcoming. But it's like the Matrix. You can't know about a Chris Mason talk until you experience it. So That was amazing, Chris. I think that the chat has been blowing up with all kinds of reaction to your talk. We definitely have time for a couple of questions for Chris. So most of it is just kind of people being blown away by things. But if you want to raise your hand and ask Chris a question directly or put it into the chat, that'd be great. I've got a couple of quick questions. So when you were showing the data, the swabbing data that you had, you made the point that you did this analysis of all the different surfaces. So you said you weren't you weren't sure if it was infectious or not, because that's one of the kind of critical questions, right? Is it actually infectious? So can you just say a little bit more about that part of it?
Yeah, that's one of the key. It's a great question. So the fomite transmission is even this little it's like the third rail of COVID testing. Like, can you get COVID? Like, I don't know, in the early days, like if you were like me, like I actually was spraying down my grocery bags when I come home, like back in March, and I was like, because no one knew at that time.
Right.
So I guess a bit of good news is every single one of those hospital samples, like we can see the virus, and you can even get the whole virus, and sometimes it's still intact. But we only tested 12, so it's an N of 12, but none of them were infectious when we brought them into Vero E6 cells in a BSL-3 lab to see, can you get them to infect cells? So far, no. Most of the data that's been published, there was a University of Nebraska study that showed it was possible about 3 or 5% of the time, maybe they'd be infectious. So in general, I think the view of the field and our own data shows that if it's possible, it's rare, and most times probably not likely. Even if you can see the virus, it's probably not infectious. But it just depends. If someone just sneezed on a surface and then 3 seconds later you walk by and you touch it and then you lick your finger, that would probably be bad. But I stopped licking my fingers after riding public transit a while ago. But so odds are low. But I think it's still not. There's some people think it's not possible, but that's already been shown by 2 groups that it's just really unlikely.
Okay, that's really great. And let's take a couple of questions from the audience. So let's go to Natalia. Natalia, you want to go ahead? I think you have to unmute yourself, Natalia.
I'm trying to go through.
Oh, there you go.
Hello.
Yes, go ahead.
Thank you, Chris, for an amazing talk. We are also, as David said, mind-blowing. I'm just curious regarding the swabbing also, because as you said, it could not be infectious virus, but it could be really important to have epidemiological data to take actions. For example, right now that most of the countries are open their airports, we are kind of tracking the viruses that are circulating in our countries, but I don't know if this kind of data, the swabbing ones, for example, in airports, could be important to track the changes or mutations and new strains that are arriving to the different countries.
Yeah, that's what we've been looking a lot at now is, do we have any evidence basically of any new mutation? So that's one really good thing you can see for vaccine development, for primer design, for ways of tracking. You need to be able to look for new mutations or any variations, and that's absolutely one of the key and obvious uses of it. But we're also looking to see, do we see other mutations that just might be popping up? So we just had cases last week in the hospital. I kind of alluded to this, is that they were coming in, they were much younger patients, and they also generally had a lot more, basically the younger patients, but the CT values were lower. And so we're trying to see how many mutations they had. Actually, I'll share my screen again. This is ongoing. So we're sequencing from the patients, but also from environmental. And we were looking at fall versus spring, and just the number of mutations we're seeing per viral genome. It's very new data. This is literally from 2 days ago. So we're still looking at this, but it seems like they're getting lower CT values or higher viral content in some of the more recent samples we have. And we're looking at this. It's still too low of numbers to be sure, but we just got another batch of samples Friday, and the sequencing's ongoing. So it's one of the advantages of doing that is for sure to be able to look at those questions. Also, sewage sampling. And sewage is probably, the virus in sewage is not really infectious, but it helps you see where is it, how much of it is. So that's also been that's interesting. 2 years ago, sewage sequencing was kind of like a weird, mysterious, frowned upon effort, but now everyone's like, well, we should probably do this. And University of Colorado is doing all their dorms. I know there's some being done in Madison, some in Racine, Wisconsin. So, poop is where it's at now. It's really, that could be like a t-shirt or something.
Poop is where it's at. Shout outs to BioBots and Mariana Matsu from MIT, who's been doing amazing work on this for years as well. All right, Craig, we'll close it out with you. Go for it.
One, Chris, thank you for that incredible presentation. I'm also a fan of just hitting people over the head with a lot of data and letting their subconscious process it. But I think that's really powerful because we live in this an incredible time where there's so much misinformation.
Yeah.
And I think as people are even skeptical, with the president of our country saying, oh, it's fine, but paying no attention to the amount of support he got, I think it just how do you see that the community coming together to support and push these initiatives along with the academic community and localized biolab communities can help to, one, deploy these treatments that are developed in a rapid response, but also to help educate people, to bring them into the fold and show them, hey, look at all this information, because I think a lot of that misinformation is done in a similar fashion where it's shown up front quickly. But how can we convey this information and help people learn how to act appropriately and how also to like understand the gravity of the situation?
Yeah, those great questions. I think on the first one, in terms of the sharing the information and about the clinical trials, about the treatments, as well as about testing methods, it's groups like this one, actually, very much like this whole community, the Bio Summit, this entire group, as well as like the GLAMP group, is basically just different groups coming together to share information as fast as they can. Because I don't know if anyone remembers, but there was an early report that maybe, there was where did the virus come from, or where there's some preprints, about whether hydroxychloroquine works, and I think it's some people were annoyed that preprints got taken down, but also New England Journal of Medicine papers got retracted. But the good thing about science, though, is generally it's fairly quick in its self-correction cycle. So generally within days or weeks, any bad paper was pulled back or retracted or quickly criticized. So I think the bedrock of science, of good science, is replication, reproducing data results. But the only way you can do this is if you share data, especially between medical systems that normally keep things siloed. So we've seen like the journals have all gone to every COVID paper has to be online and accessible, and we've seen them reduce fees and get things out faster. So I think I'm hoping, we've even seen airlines take away change fees. Like I'm hoping there's been some things maybe will go a little bit to a better place after this, to say, well, maybe if people share data faster, that's actually a good thing. Maybe if it's an obscure biophysical trait you're studying that's not related to a pandemic, maybe that doesn't have to be open access media, but we could make an argument that all journals should, for all time, make anything related to public health or an active pandemic always should be open access, or and maybe it'll hopefully stay that way. So I think that's one thing. And then just the misinformation, it's just keep being a good scientist and keep questioning things and saying, are you sure about that, or what's the source of that? Has that been replicated? So just be critical as we always are. And then also talk to your weird uncle at family functions, or your grandma who says, when people say, oh, I hear it, if you've already had the cold, you can't get COVID. You're like, that's just not true, and like, you just always don't be bashful about politely calling people out on bullshit, because it's everywhere. Yeah.
Okay. We have a Slack channel, #words of wisdom. That is a good one to probably put in there on calling out the bullshit. So thanks for sharing that, Chris, that nugget in particular. But I think that's also a great segue into our next session after our break, which is on science communication.